For the Module 2 component of Section A now of the 2018 biology paper, we're looking at the seed type in pea plants and the fact that it's determined. Hold on. Alright, so it's determined by a single allele with the capital R being for round and common R being for wrinkled, and 3 pair of chromosomes are depicted. And what we're supposed to do now, right, we'll see that X here describes the loci. Right, so we're supposed to show in A homologous round seed, so homologous recessive, homologous, well, that would be homologous dominant, homozygous rather, homozygous dominant. B should be homozygous recessive, and C should be heterozygous. So we should see that there. So therefore we're gonna have large R, right? Then have small r. We're gonna have a large R, small r. Right? And they should be on the 2 sister chromatids there, because 2 different alleles, right? So we have that there. So they generally spread across 2 different chromatids, right? There's another expression in which it could be at different points on the same chromatid, right? But this is probably a safer way to answer the question, right? So we have that there. Really simple. 3 marks, honestly, right? 3 free marks. All right. The next question: draw a Punnett square, right, to illustrate the crossing test involving a P variant which is heterozygous for the seed type, right? So we're looking at generally how I interpret this question. We're looking at involving a heterozygous type. So it could be heterozygous with heterozygous, could be heterozygous with homozygous dominant, it could be heterozygous with homozygous recessive, right? We did heterozygous with homozygous recessive yesterday, so let's use heterozygous in this case, right? Heterozygous, right? So we're gonna have R, small r, lovely, right? And what would be, I'm not sure if I'm moving too fast, right? Right, but what would be the ratio for the phenotypes now? We know that, right, these would be round, right? Therefore, the ratio would be 3 to 1, right? Lovely, all right? And then I guess you could speak to this being round and that being wrinkled, right? You could speak to that. Lovely, moving on. What is distinguished between a gene and an allele? We know the gene is a specific sequence of genetic information which codes for a specific strand of RNA, which codes for a specific protein, right? So it helps to provide that genotype for the expression of a phenotype. While the allele is a different flavor, I guess. You don't write flavor on the paper, right? But I always think of it as a different flavor of gene, or it's a different type of gene or variant of the same gene. So gene codes for a specific characteristic, right? But the allele changes the type of characteristic, or what characteristic does that gene give. So the gene could be for eye color, but allele could be, let's say, a gene for brown eye color versus blue eye color versus chestnut eye color, right? So you could look at that. So a gene codes for a specific phenotype or specific characteristic. One allele is a different form or variant of the same gene. Lovely. Let's move on. Right. Still on this area now. Now I had some contention yesterday as it relates to this diagram. Right. We're supposed to be looking at the stages of mitosis within these cells here for D, E, and F. Right. Person's assumption. Right. Well, it's generally assumption for everybody, to be very honest. Right. Because biology is left up to interpretation many of times. What people were saying last night is that D was prophase, E was anaphase, and F was interphase. But what I was saying is that D is interphase, right, and F is prophase. Right, my rationale for that would be because what we're looking at generally is the condensation of the chromosomes. Within D, it's more thread-like, right? It's less condensed. So I would think that would be interphase or even early prophase before the condensation of any of the chromatin into chromosomes. While F, you can clearly see condensation of chromosomes. I would think that F would be more into the prophase. So I would describe D to be interphase and F to be prophase. Right, but we were having a little bit of a contention over whether or not that would be so. But I think that would be the best treatment to the question. Right, So D interface, E anaphase, and F prophase. But E, nobody had issues with E. We see the chromatin dragging the genetic material to opposite sides of the cell. We know that E would be anaphase. Alright, within that meristem. We're called to draw these, so you guys should be able to draw these structures, right? And I think you guys did a lab on mitosis as well, so you'd have drawn them or constructed them. So everything there should be lovely. Everything there should be fine. Alright? That's the end of that section. And we move on to the module 3 component, yes? Alright? It's module 3 component of section A. Yes, the module 3 component of section A.